People were worried about the killer bees, right? And you might remember the Saturday Night Live, right? John Belushi, the killer bees, right? Everybody was all worried about stuff like that.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I am Kim Klinowski, your host for today's episode. What do honeybees, elk, ticks, alligators, and parasites have in common? One thing is that they all have been studied by today's guest.
Our guest today is Dr. Travis Glenn, professor and head of the Department of Environmental Health Science and director of the Institute of Bioinformatics. He's also affiliate faculty in the Department of Genetics and the Interdisciplinary Toxicology program. Travis, thanks for being with us.
Yeah, thank you.
So Travis, one of the questions that we ask all of our guests is, can you tell us a little bit about your pathway to science?
I can. My question is, how long do you want that journey to last?
Give us the highlights,
Okay.
or maybe your favorite parts.
All right. So I'm a Midwestern farm kid. I have known from a very young age that I was likely to go into science. I've always had an affinity for it. I first started programming computers in the 5th grade when my elementary school principal built his own computer back in the days when there wasn't a ROM chip. So you had to take a cassette player and play 30 minutes or more of, noises that sounded like the old modems, right? Before you could actually type a command on the keyboard so the computer could understand you.
The good old days.
That is true. And so I was a computer nerd pretty early back in the days. I remember my father taking me to Iowa State University for the cluster computer, and you had the hollow floor with the noises of the hollow floor that you could hear, and then the print cards and things like that. So I had the old print cards. But I have 3 older sisters, and my second oldest sister in particular was very concerned that I would be too much of a nerd. Okay. And I mean, she was by far the smartest child that my parents had. She was the only person that I knew that was, valedictorian and athlete of the year. Oh. Right. So, I mean, very accomplished person. She wanted to make sure I wasn't too much of a nerd. Mm-hmm. Okay. So I did lots of hunting and trapping and fishing and all of the outdoor activities. So that was kind of the competing thing was I going to be, an electrical engineer and, deal with computers, or was I going to follow my passion in the outdoors? And so I had the opportunity to become one of the early scholars for the Iowa State University's Ag Biotechnology Scholars, the inaugural, guinea pig class. And so that was an opportunity to use science in the field of animal ecology was my major, which is sort of fish and wildlife biology for folks who knew they wanted to go to grad school. And so that really opened up opportunities for me to learn about DNA and how it could be manipulated. And I embraced that. I loved that. But I thought, well, what that would set me up for is to be a really good regulator. So I'd understand the technologies and how you could write laws to ensure that they were used safely for society. And my senior year, I took evolution, and the instructor there taught me so much about how we could actually use molecular techniques to understand wildlife. And it was like, whoa, right? So
that sounds like a great class.
Yeah, so I was able to take my passion for, I was a park ranger as my summer job for 3 years as an undergrad. And so I could take that passion and combine it with science. And so then I went to the University of Michigan and studied the reintroduced elk population in Michigan in the basement museum. So I was working in the basement of the museum. I had this vision of working on the elk population that was in the state. I'd worked with hunters, collected samples from them. But I didn't have any practical knowledge of how to actually assess genetic variation in these wild organisms. So I worked with a postdoc there, Deborah Smith. She was working on honeybees. And so the exchange was she would teach me how to do protein electrophoresis, and I would do a bunch of her bee samples for free, as free labor. in exchange for learning the techniques and then access to the equipment so that I could run my samples on them.
Mm-hmm.
And so she was very gracious about that. And one of the first sets of samples that we worked on were honeybees that had been collected by the USDA in southern Texas. And the question was, had they been Africanized? And so we were among the first people to be able to genetically verify that there were populations of honeybees in South Texas that had the alleles that showed they had the genes that had been brought from Africa.
I remember that story, that bees were becoming Africanized, which I think basically meant they were more aggressive.
That's correct.
Yeah.
Yep. So that was a big thing, right? And there were, movies and blah, blah, blah. People were worried about the killer bees, right? And you might remember the Saturday Night Live, right? John Belushi, the killer bees, right? Everybody was all worried about stuff like that.
So this is interesting to me too, because when I think of, you use the word museum.
Yep.
Right. But this is not like the Natural History Museum per se, or is it?
It actually is. So we were in the basement of the Natural History Museum at the University of Michigan.
Oh, gotcha.
And so one of my committee members was a curator there, as well as a faculty member, a mammologist. And I actually went from Michigan then to the Smithsonian. So my PhD work was at the University of Maryland, but I did my work at the Smithsonian in the Museum Support Lab in Suitland, Maryland. And so if you've ever seen Raiders of the Lost Ark, right, that last scene where they're taking in the Ark of the Covenant, right? Going to this huge thing. Okay, they shared with Steven Spielberg the blueprint for the pods, the storage pods, where the labs were where I worked with him, and they CGI'd that in because they hadn't finished construction yet. But they were just months apart from so they knew what it was going to look like.
And it's really that big now.
And it's as big as a football field, 3 stories tall. The first pod where they're showing, what that scene is, it's mostly open with things stacked up. Now, if you go to the other pods, they have racks in there. So if you're looking for the elephant heads from Teddy Roosevelt, when he went out and collected things for them in Africa, those are in pod 3, right? So
this is fascinating to me. It seems like the coolest job in the world. But I guess the other thing, so are these museums still actively collecting samples?
Yes, absolutely. Now, and it's a variety because it depends that some of the animals and plants that you're most interested in are endangered, right? And so there's a trade-off between knowledge and caring for the populations of those organisms.
You don't want to kill the last dodo.
Correct. That's absolutely correct. So there, for sure, when you're dealing with endangered species, there's a lot of care given to, should we just photograph it, take a blood sample, do things like that, versus do I need to have at least one type specimen so that I know what it is and can describe it in detail, right? And so there's a trade-off, right? And you want to make sure that you're not further endangering these populations that are, very limited.
So one thing, and I think it relates to your talk about the Africanized honeybees, a lot of your work involves genetic markers.
That's correct.
Developing those. So maybe you could give us a little explanation of what a genetic marker is and how you develop that.
Yeah, so a genetic marker you can think of as a unique piece of DNA that's shared among organisms that have an ancestry, a shared ancestry, that you can use to define that group, right? And so it's a little bit of looking for a needle in a haystack because a lot of genomes are very big and you're trying to find the things that are shared by ancestry and not unique to each individual, but they aren't so broadly shared that they go too deep into that ancestry. So the trick is sort of finding at what level do I want to find that ancestry? Is it a species group? Is it a family group? Is it a group of species, a genus of organisms? What is that?
So I know the types of genetic markers that people use have changed over time. Now, has it gotten to the point where we just do whole genome sequencing on everything and call it a day, or
It depends on how many individuals you're trying to assess. That's probably the biggest thing. And over what period of time you'll have those samples. Currently, it is usually most efficient to sequence the whole genome of a small number of individuals, find those chunks of DNA that are of most interest to you, but then if you're going to assay lots and lots of individuals, then convert that sequence into a different type of marker system. And it might be a polymerase chain reaction where we're able to amplify DNA from a tiny starting amount of material, or it could be other assays. It depends on, the type of organism, how quickly you need the results back, how many assays are you going to try and bring online, things like that.
So I think I see kind of a connection between your early interest in computers and development of tools, because a lot of what you've done is developing genetic tools. Am I accurate there?
Yeah, absolutely. So again, a story people might find interesting is that they think you can find everything online now, right? So the Smithsonian has a great museum, a website where you can go in and get lots of videos and things. I was at the Smithsonian before they were connected to the internet. Okay. Which is, wait, what? Are you kidding me? Okay. So, and we were there, in 1991, there weren't web browsers. They didn't exist. The internet existed, but there was no such thing as a web browser. So all of that was a transition, during my PhD program of being there while the systems were changing in these very radical ways.
I recall as a graduate student, we got the package in the mail from GenBank, which was 5¼ inch floppy disks. And you could look up a name in an index disk, and it would tell you which disk to go to look for the available sequences. Yeah, I know.
You guys are totally dating yourself, and I'm just letting you go, and I'm just sitting here and not saying a word. That's my stance. So speaking of, we've talked about the fact that you're really interested in developing genetic tools. And when I was doing my research on you, I noticed on your lab webpage, it says the goal of your lab is to develop new genomic tools to investigate processes in evolutionary biology, which we touched on, solve problems in environmental health, and infectious disease ecology. And that is a lot. So I'm not sure that I could personally take on more than one of those things. So are you simply interested in a lot of things, or are you using some of the same techniques to address some of these problems, very different problems?
Yes.
Yes, right. To all.
Yes. And so one of the problems with developing marker systems is that it can be a good bit of work that only applies to one system. Mm-hmm. And so I spent a significant portion of my youth at the Smithsonian because I knew this technique that could be used to develop species-specific markers or to do multiple paternity assays and determine breeding systems of all sorts of different organisms. But you had to develop those markers for each species. And so that made me popular, which was great. I got to meet a lot of people that are, friends to this day. But it meant that we did a lot of work that you couldn't extend to other things. Mm-hmm. So when I moved here from the Savannah River Ecology Lab to Athens, we were right in that transition of next-gen sequencers. So the 454 was just coming online. Illumina instruments were just coming online. The ability to sequence a whole genome for less than millions of dollars was just becoming available. And so one of the grand challenges that one of my mentors at the Smithsonian had asked me to think about is how could you develop a marker system that would work so that I could determine the phylogeny of all birds, right? There's 10,000 birds. I want to do them all, and I want to be able to do an apples-to-apples comparison across all those species. And this is sort of a famously hard problem, especially as you get to orders of birds, because they diverged almost simultaneously at the KT boundary when all the buck dinosaurs went extinct. So they get to that point, and then they're all equally divergent. So it's very difficult to tease apart which one is related to whom the most, right? And different parts of their genome will give you different signals. So you can't just look at 1 or 2 markers, and you can't look at just 10 or 20 markers. You need hundreds of markers, if not maybe a few thousand. And so I sort of had this notion that if we could find anchored points throughout the genome that would be consistent among all of those species, then we could do an assay using these brand new instruments to sort of look at these, thousands of markers simultaneously in a very efficient way. And so I was able to work with a senior PhD student in Warnell. His name is Brant Faircloth, and some other folks that I know at LSU and have known from grad school, and other folks who were in my lab over the years. We sort of had this confluence of folks who are coming together around this technique, and we spent some time and energy and developed the ultra-conserved elements. Are these things where if you take a whole genome of 2 organisms and you look for the bits that are exactly identical. It was sort of right at that time when you could do a full genome alignment of a human and a mouse, and they were just bringing on board the first bird, and we were doing whole genome sequencing of the first lizard. And so we wanted to be able to develop those marker systems that would work the same across a huge group of organisms. And so we spent several years getting that to work right to solve this problem of how do you use one marker system across 10,000 species? And actually that marker system worked better than what we imagined. It works across all tetrapods. And so there's, 20,000 species that you can use this exact same marker system from. And so that really has been my goal then, is to take that same idea and now apply that in the infectious disease world, right? So can we develop these marker systems that would work for Cryptosporidium or for other organisms that we're interested in?
Well, let's since you mentioned it, let's move on to crypto.
When we say crypto, we don't mean cryptocurrency.
No. Good point. Because as a parasitologist, parasites are close to my heart, and fortunately, in the case of Cryptosporidium, not close to my gut.
Yeah.
But how did you get involved in that project?
So that basically was all working with Jessie Kissinger. So she is an expert in this system. It is a group of organisms that are not very well known. There's a few that are known pretty well, but it's a very diverse group of organisms. They're small, they're hard to work with. The pool of experts in the world is relatively limited, but it's great people doing really important work. These organisms are important in the United States, so that people understand this organism. If you go to the pool and they're closing down the pool, it's probably because Cryptosporidium have gotten into that pool because bleach doesn't kill the oocysts, the little eggs that transmit this disease to people. And so we do testing for it in the United States. If it gets in a water distribution, it's really bad. The most famous example is when it got into Milwaukee.
Oh, yeah.
And 400,000 people got ill because of this.
If you thought toilet paper was hard to find during COVID, imagine a whole giant city with everyone
getting crypto. Yes.
Getting crypto.
That's ungood. And so would you like to describe the symptoms of those people?
Well, chief among it is diarrhea, which can be bad enough that you can get severely dehydrated. I mean,
and be hospitalized.
Right.
For that.
Right.
That's correct. Yeah. And so folks who are immune compromised or babies or elderly, it can cause them to die. It's pretty rare that it causes death, but it's pretty common in those sensitive populations that it really is a bad thing. It can stunt the growth of children. And so you can imagine in places in the world where getting clean water is a problem, that crypto is more common, and it's a real public health problem. And so I'm a member of the College of Public Health at the University of Georgia, looking for more projects where I could apply this tool set in a way that would be helpful to solve a public health problem. And so Jessie, I knew from her work that she was working on crypto and was engaged with the community. And I felt like this might be a place to take a tool set that I had and apply that in a way that could be helpful.
So what questions are you asking?
So there's a huge variety of questions that you can ask once you have the tools up and running.
Mm-hmm.
And so it ranges from things like, are these different outbreaks of crypto caused by different species of crypto? Are they, if it's an outbreak and it's different individuals, are they all from the same clone?
Mm-hmm.
How is it that if you're an individual, especially in one of these places that has limited access to clean water, and you get crypto multiple times, is that a different strain of crypto each time? Or are you sensitive? Do you regain sensitivity? How does that work?
Mm-hmm.
So there's lots and lots of questions that you can ask and answer, but you're limited by the number of genetic markers and can I use all samples available to me, right? So one of the things that you might be able to do is if you get a large enough sample of poo,
Mm-hmm.
right, then you can isolate the crypto, the oocysts from that poo. But the problem is it causes explosive watery diarrhea. So getting an actual large sample is more challenging than you might imagine. So one of the techniques is to how little of starting DNA can we work with to still come out with multiple markers from many different parts of the genome. And so where we are currently is that we've developed the system to capture the whole genome.
Mm-hmm.
We're looking at several, hundreds of samples that are being screened from thousands of samples that our colleagues from around the world have sent to be able to get a better survey of the full genome to understand now can we hone that down to a smaller set of markers that would be similar to what we used for the birds to be able to look, really scale it up and look at very large numbers of samples at a more affordable cost.
One other question about cryptosporidium. Is there any unique challenges in working with that genome?
There are. So it's a eukaryote, right? So it has longer sets of repeats than you might encounter in something like cholera or other bacteria. So that makes it a little more challenging, but it's also the way in which it reproduces. You can't really get down to a single haploid or a single diploid individual, because by the time, if you're dealing with the oocyst, there's 4 cells that are in there, right?
Okay.
So it's sort of, and if you're dealing with a sample from a person, you're sampling an entire population of what was in them. And the question is, how many individuals were they infected with originally that gave rise to that population?
So I know David loves to talk about parasites and could do it all day long, but I kind of want to shift the topic a little bit. I believe you're also interested in antibiotic resistance.
That's correct.
So I want to spend a little bit of time talking about this because I think you're approaching antibiotic resistance from a unique perspective.
So again, my interest is sort of in figuring out how are genes distributed around the world and how are human activities impacting those distributions. And so antibiotic resistance is something that we know is a public health issue. It's growing because if you use an antibiotic, you've lit the fuse on how long you're going to be able to use it in the future. My interest is in knowing how long is the fuse? Are there things that we can do that can reduce the amount of antibiotic resistance in the environment so that there are fewer that get into people and then cause us health problems downstream? And so one of the ways, there's lots of ways you could look at this, right? You could sample the environment and ask what's there.
Mm-hmm.
You can also sample people and ask what's in them.
Mm-hmm.
The thing that most people are sampling for microbiome is their poo.
Mm-hmm.
Okay, so again, I was able to work with colleagues here at UGA. So Liz Otteson from microbiology, Aaron Lipp in environmental health science, Justin Ball has also been part of this process. But the main study that we did was looking at healthy individuals in Athens, where people were willing to contribute a poo sample. So I've said, if you pooped in a paint bucket and got $50 for it, thank you. I appreciate your contribution to
Any of our
science.
listeners contributed?
It's possible, right? So the things that we did, right? So Aaron's group actually, bless their hearts, they took the poo sample, right? And then they plated out bacteria, and we selected for E. coli strains. And then from some of the E. coli, we did whole genome sequencing to understand what's in them. We also, for up to 48 colonies per individual, they tested them for a range of antibiotics. So we know the phenotype for antibiotic resistance. So doing different levels of antibiotics for different kinds of antibiotics. And then we did whole genome sequencing for sort of representative individuals. And then we did community microbiome assays, both doing shotgun and 16S. So looking for a bacterial gene and asking who's there. If you do shotgun, that's whole genome sequencing from everybody who's present. And then we enriched those microbiomes, the shotgun microbiomes, for the resistome. So all of the genes that were in the CARD database, so a database of all resistance genes, not only to antibiotics, but also to metals and other things. We have the same kind of assay that we did for the birds and that we have brought on board for other systems for crypto, is that we enriched that community for the genes that cause antibiotic resistance or resistance to other things that are related to that, right? And then we've done surveys, and then we're characterizing the communities inside each individual, then for lots of individuals, and then we're asking, what are the lifestyle sorts of things that people who have higher amounts of antibiotic resistance might share in common? What are the risk factors that they might have?
Oh, it's fascinating.
So the resistome,
Yes.
that's another ohm. I
It
just
is another
learned a
ohm.
new ohm because it used to
Yeah,
be
the resistome.
genome and
Oh,
transcriptome.
You're dating yourself again.
So I guess the resistome could be pathogen specific in a way. So you'd have a different resistome for Cryptosporidium, say, than you would for bacteria or viruses.
Right. And different bacteria will host different genes in them, right? So some of those genes are relatively specific to groups of bacteria. Others are quite broad and get shared widely. And so just knowing that something is present doesn't always tell you who had it. You know what I mean? So we have a profile of who are the common players, and we have a profile of what are the common things that they're carrying that we're interested in. But merging those together, the only practical way currently is to spend a lot of money on sequencing, right, for long reads.
One thing I found interesting was that apparently, and I did not know this, but some of the genes that provide resistance to heavy metals also provide resistance to some antibiotics?
Yes, absolutely. And again, we've known this for decades, and that goes back to a study that I did with colleagues. So Ramunas Stepanovskis is probably the main person who's the lead author on the papers that came out. He's now a faculty member at Bigelow. They do single bacteria sequencing, whole genome sequencing from individual bacteria. So he has a whole operations. If you get the opportunity to talk to him, you should do that. He has really cool stuff. But back, in the early 2000s, in the early aughts and mid-aughts, we did a study where Javon MacArthur was working on stream systems on the Savannah River site and looking at antibiotic resistance in the microbes there. And he noticed that some of the stream systems that didn't have any input from cattle or people had really high resistance rates. And he's like, what could be doing that? Well, possibly the metals from site operations could be driving that. And so we changed the system a little bit by looking at coal-fired power plants. And we looked at the aquatic system going into the power plant, so the input pipe right at the input, and then looking at the output after they had been exposed to the ash and the fallout from the ash. So they were exposed to metals, and you can test the metal levels at the input and output pipes. And across a broad spectrum of metals in general, they go up by about an order of magnitude.
Okay.
Right? So there's sort of 10, maybe 100-fold increase in a broad spectrum of metals. And so you wouldn't be surprised that if you test bacteria exposed to that at the end, you take those bacteria and you challenge them to more metal, that the community would be more resistant, right? They would be able to survive. They'd be able to tolerate higher levels of metals, right? That's totally predicted. We showed that that's true. But if you take antibiotics and challenge them with antibiotics, they're also much more resistant to antibiotics, including frontline things like Cipro, right? So things that you really don't want to have widespread resistance out in the environment. Okay, and so we showed that for 3 different power plants, 2 in South Carolina and 1 from the Midwest. And again, I don't think that's specific to those systems. It's just showing that if you have this common human activity that is increasing metal concentrations in this aquatic environment, the unintended effect is that you are, elevating these mechanisms that cause resistance to metal that also cause resistance to antibiotics.
I find that fascinating because when I think of antimicrobial resistance for bacteria anyway, I'm thinking overuse or misuse of antibiotics.
And that's what we've all been trained to think about, right? And the most important thing is take your full course of antibiotics. Don't take antibiotics if you're not sick with something that they could actually treat, right? If you have a viral infection, it is not going to help you to take an antibiotic.
Right.
So, right, and those are true, right? We don't want to diminish the importance of good public health messaging, but that's not the whole story, right? I mean, that's sort of the Paul Harvey and the rest of now for the rest of the story.
So one other thing, and maybe we can close on this. We've talked about sampling honeybees and elk and birds and Cryptosporidium and bacteria, but there's one other critter that you have famously sampled. And actually, and I'm going to lead in with a story from a friend of mine who I was talking to. So Bill, if you're listening, this is from you. He told me this story about getting invited to go out and catch alligators with this faculty member from UGA. And he swears that one of them was bigger than the boat. So my first question is, was that bigger than the boat?
It may have been a 12-foot alligator and a 14-foot boat.
Okay, close enough.
Yeah.
I don't know that I ever caught one that was bigger than the boat, but as big as and bigger than the space in which you normally would fit. That is true.
That's about 12 feet longer than the alligator I would want to mess with.
Exactly.
So you were catching them to get blood samples?
That's correct.
And then doing what?
We were doing a couple of things. So we were interested in measuring the metals that were inside them, in particular mercury is a metal of concern in a variety of alligator populations. It's not a really big concern on the Savannah River site, but in other locations, it's a bigger issue. We were also looking at paternity and parentage of the alligators and their offspring on site.
It's a legal thing.
So the thing that we were particularly interested in is that we know that these alligators have been exposed to radiation. They have it inside their bodies. It's a very small amount. If you consider the amount of radiation that you get from a dental x-ray, and you take that and you spread it across a year, that's approximately how much additional radiation these alligators were exposed to from eating this radiation from a leak back in the 1950s, okay, or early 60s. So it's in the environment there. It can be stirred up by different site activities, but you can measure it, right? Because you can measure vanishingly tiny amounts of radiation, you can measure it above the background rate. But we were interested in just the breeding system of alligators in general, which is how I got to work on alligators in the first place, is that my PhD advisor's PhD advisor was interested in just, do alligators have multiple paternity? Birds obviously do. They have extra pair copulation, all of that stuff. So the breeding system of them was unknown. And so I developed this set of markers that was PCR based. That was the same thing that I was doing at the Smithsonian, right? So I was in that period of time, I developed markers for alligators so that we could answer this question. And so once we had those markers, then we could ask lots of other questions. And one of them is, because the mutation rate for these particular markers is so high, you can measure it between you and your offspring, right? So, or you and your siblings, or you and your parents. That is a known thing that you can measure. But it's not huge, right? It's like 1 in 1,000. But you have thousands of these markers in your body. And so if you have enough offspring and enough parents and enough, markers, you can directly count those mutations between parents and offspring. And so the question was, is there a higher mutation rate, right, that's caused by this radiation exposure, this heavy metal exposure between parents and offspring for the top level, carnivore on site? Is that causing an issue? And we were able to show that it's not, it's not an issue. So it's a public assurance, right, by outside scientists. We're not paid by the government, or, the message is ours. The data are, as genuine as we can get them to be, to assure the public that, yes, there are, you can find 4-legged owls on site, but that's probably not a radiation thing. That's just every 1 in a million owls has 4 eggs, or 4 legs. So.
Well, I think one last question that we're interested in is, it's hard to ask this question after that story, is so when you're not trapping alligators, what else do you like to do in your free time?
Oh, well, so my alligator days in the field came to an end as I had children.
Okay.
So I had many more hours in the lab and writing, really, once the kids came along.
Okay.
But my youngest child is 2 months from turning 16, which, as every parent knows, that is sort of liberation day. You're no longer Ubering, right? That comes to an end. So my wife and I are in active conversations of how are we going to readjust as we now have more and more time available to go do things like that. But yeah, back in the day, the question people were like, what do you do for work? I catch alligators. What do you do for fun? Same thing. What do you do? Right. So hopefully I'll get to spend a little more time doing that in the future. But, going hiking and things like that with the kids, that has dominated our life.
Links to additional information about Dr. Glenn's research can be found on our Instagram account, which is at PPPPodcastUGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Justin Nemitz, for research, editing, scheduling, and keeping us on track.